Good morning, everybody, and welcome to this immunology pill on antigen and virus-specific T cells. Today, I will discuss the importance of antigen and virus-specific T cells and introduce to you our workflow to analyze these very rare, but at the same time, very important cells. So enjoy. So here you can see today's agenda. So first, we will see what antigen-specific T cells are and why we need to study them in COVID-19. Next, I will talk about some key challenges that arise when working with these rare T cells. And then we will look at our workflow solutions for the analysis. So let's start with a look on how we can define antigen-specific T cells. So of course, all T cells are in principle antigen specific. However, here we talk about T cells that were activated before analysis by their specific cognate antigen and not via a polyclonal stimulus, for example, CD3 or CD28. The initial frequency of this antigen specific T cells before stimulation is very low. However, through the activation antigen specifically, only T cells of a given specificity are activated. So they upregulate certain surface markers, start to secrete cytokines, and also clonally expand. And of course, antigen-specific T cells play a key role in the immune response against all viral and bacterial infections. So therefore, the analysis is highly important in both basic and clinical research. For example, in the research on infectious diseases, tumor immunology, or also vaccine development. So why do we need to study these antigen-specific T cells, or also in this case, virus-specific T cells, especially in infectious diseases like COVID-19 in more detail? So first of all, because we need an accurate picture of SARS-CoV-2 immunity and immune responses. So for example, research into COVID-19 has revealed that about 10 to 30% of patients do not produce detectable levels of SARS-CoV-2 specific antibodies. And that especially in asymptomatic and mild cases, antibody titers are really low and also decline rapidly. However, in contrast, it has been shown that SARS-CoV-2 reactive T cells are also generated even in the absence of virus-specific antibodies. So for example, in this study by Sekine et al. Published in 2020, you can see that T cell responses were detected more frequently compared to virus-specific antibodies. So you have the T cell responses here on the right and antibody responses on the left, particularly in cases of mild infection, which you can see here in this pinkish, where you have like a high detection of specific T cells in mild cases. Importantly, the analysis of SARS-CoV-2 specific T cells can also be used to monitor long-term immunity, be it after natural infection and also vaccination. In particular, in regard to vaccines, it will be very important to study the implications of cross-reactive T cells for the design and analysis of all these upcoming trials that investigate COVID-19 vaccines. And last but not least, the SARS-CoV-2 reactive T cells could also be used as a treatment options, for example, in adoptive cell therapy. So how can we analyze these antigen and virus-specific T cells? So, well, generally there are two methods that can be classified in two main categories. So first, there's the direct analysis of T cell specificity, and here the T cell receptor is labeled with so-called peptide MHC multimers to directly identify antigen-specific T cells. And this method has certainly the highest specificity for the identification of antigen-specific T cells. And second, there are the so-called indirect analysis methods, and here more functional parameters of the T cells, such as the expression of certain activation markers or cytokines, but also, for example, cytotoxicity or proliferation can be analyzed. Suitable activation markers that could be analyzed are, for example, CD69, CD137, or CD154, and cytokines like interleukin-2, TNF-alpha, or gamma interferon. So these methods really provide T cell subset specific information, and they are also completely independent of the used antigen for stimulation or the MHC status of the donor. Most importantly, however, the biggest advantage is that all relevant information can be obtained using flow cytometry-based assays. So what are some of the key challenges that arise when working with this very rare antigen and virus-specific T cells? So as briefly mentioned before, low cell frequency is definitely one of the biggest challenges for reliable analysis. So all the more, it is important to optimally prepare the starting material without losing the cells in the first place. Furthermore, to ensure higher cell numbers, the cells can also be pre-enriched magnetically or via flow sorting. Another common problem is insufficient stimulation. And here, only by using really powerful reagents and optimized protocols, the cells can be efficiently stimulated. Furthermore, the analysis is often hampered by high background staining, just because these cells are actually so rare. And here, reagents for specific labeling and sensitive detection methods are really important. And finally, there's often a rather slow translation of early findings into the clinic. And here, Miltenyi Biotec actually offers a large portfolio for translational research that could help you to speed up this process. So to efficiently isolate, stimulate, stain, and analyze antigen-specific T cells, we combine different technologies over an entire workflow to ensure a successful analysis. And of course, here, we think the right product combination is actually the key to success. So now let's take a look at the complete workflow for the analysis of antigen and virus-specific T cells. So of course, successful experiments always start with sufficient sample preparation. So first, the T cells need to be isolated from a donor sample, and this could be tissue, blood, or PBMCs. And of course, for tissue dissociation, our GentleMax tissue dissociator is a really good choice. Next, the cells are then stimulated with the antigen of interest. And please note that antigen-presenting cells need to be present to efficiently activate isolated T cells with a certain antigen. When working with PBMCs, for example, this is already the case. However, with isolated T cells from tissues, antigen-presenting cells would have to be added separately. Following the stimulation, only the antigen-specific T cells will get activated, and the cells can then be stained intra- and extracellularly to assess the phenotype and also analyze cytokine expression, for example. Before analysis, the cells can then also be pre-enriched magnetically. And then finally, the cells will be analyzed, for example, by flow cytometry or sorted further on a cell sorter. And you can find a good overview of the whole antigen and virus-specific T cell workflows, including protocols and product solutions, on our application websites by following these links here on the bottom. So now let's take a closer look at our products for antigen-specific in vitro stimulation. And here, of course, you can achieve really efficient antigen-specific stimulation with our Peptivator peptide pools. So these Peptivators efficiently stimulate both CD4 and CD8 positive T cells, and we actually cover an extensive portfolio of virus, tumor, fungi, microbiota, and even autoantigens. The peptide pools always consist of 50 mer peptides with 11 amino acids, covering the complete sequence of the respective antigens. And our peptides are actually already available in research, premium, and even max GMP grade, which allow a rapid translation of early findings into the clinic. And they are also available in a very convenient 96-well format for high throughput applications. And of course, we also offer several Peptivators specifically for SARS-CoV-2 specific T cell activation. And here, for example, we have SARS-CoV-2 Peptivators directed against the spike. We actually have 3 different spike Peptivators, as well as Peptivators for the nucleoprotein, as well as for the membrane protein. And then most recently, we also added the so-called SARS-CoV-2 Peptivator Select, which actually contains all immunodominant epitopes of the whole virus proteome. And we also already released SARS-CoV-2 Peptivators for the new mutation variants that were originally detected in the UK and in South Africa and are now spreading worldwide. So now we will take a short look on tools that can be used for the pre-enrichment of antigen virus-specific T cells following the in vitro simulation. So of course, flow cytometry is still the most widely used method for the analysis of these rare cells. However, even methods like flow cytometry have its limits and challenges, especially as antigen virus-specific T cells can be extremely rare. And this was already illustrated in a very nice publication by Bacher and colleagues in 2015, where the authors state that despite its high sensitivity, flow cytometry is always limited by the number of events that can be acquired, as well as the staining background, which is typically between 0.01 and 0.1%. So this really restricts the analysis of cell populations with low frequencies. So although in most instances, actually modern flow cytometers are sensitive enough to detect at least some cells, there might be certain conditions where hardly any specific cell can be detected. And here you can use our Max microbeads for pre-enrichment. And furthermore, to overcome high background staining and enable a high resolution in flow analysis, our recombinant reaffinity antibodies combined with the virodice will provide you with a high specificity and superior lot-to-lot consistency, as they are ideally suited for the analysis of rare cells. And indeed, when you look at some results generated with the pre-enrichment of, for example, CD154 or CD137 microbead kits, you can see the results are really nice. So both markers are actually upregulated on CD4 or CD8 positive T cells after antigen stimulation and are therefore ideally suited for their pre-enrichment. So in this example, PBMCs were stimulated with a CMV peptivator or left untreated. And then the antigen-specific CD4 positive T cells were successfully enriched using the CD154 microbead kit, whereas CD8 positive T cells were enriched with the CD137 microbead kit. And the isolated cells can then be used for any downstream application, like, for example, TCR sequencing. And if you want to know more, you can also have a look at our very nice tech paper, which illustrates the technology in much more detail. So furthermore, I would also like to point out a cell separation product that is absolutely unique. And these are the so-called cytokine secretion assays. So cytokine secretion assays actually allow viable enrichment of cytokine secreting T cells from mouse and human samples. So the principle is actually rather simple. So first, secreted cytokines are immobilized on the cellular surface by the so-called catch reagent. And then in the second step, the immobilized cytokines are then labeled with fluorophore-conjugated antibodies called the detection reagent. And by that, you can detect cytokine-secreting living cells without any intracellular staining. And then in an optional third step, the labeled cells can be magnetically enriched with the help of anti-fluorophore magnetic beads and then used in any downstream assays. And these cytokine secretion assays actually come in two versions. So for flow analysis only, we offer various detection kits, whereas for enrichment and analysis, we have several enrichment and detection kits. And we also generated some exemplary data using these detection and enrichment kits, and they work very efficiently to enrich, for example, viable virus-specific T cells. So here, PBMCs of a CMV positive donor were stimulated for 4 hours with CMV lysate or CMV peptivators, and then the stimulated cells were then stained and isolated according to the secretion of TNF-alpha in the case of CD4 positive T cells, or in the case of CD8 positive T cells, they were enriched for gamma interferon using the respective enrichment and detection kit. And as you can see in both the stimulated samples, a very distinct population of cytokine secreting CD4 or CD8 positive T cells could be specifically enriched. So, also in some cases, we strongly recommend to enrich the cells before analysis. In many cases, you can also directly stain the cells for flow analysis immediately after antigen-specific stimulation. And here I want to highlight two of our new products, the SARS-CoV-2 T-cell analysis kits, and we actually offer two different versions of the kit. So one is optimized for human PBMC samples, and the other one is for human whole blood samples. So what were the kits developed for? Well, they actually enable a reliable detection of SARS-CoV-2 reactive T-cell responses using flow cytometry and allow analysis of CD4 and CD8 positive T-cell activation in various COVID-19 donor samples. And they can be used for monitoring of SARS-CoV-2 specific T cells after infection or vaccination, and for the research of infection and vaccine-specific T-cell responses to better understand COVID-19. So what is actually in the kit? So while the overall components of both kit variants, so the PBMC and the whole blood version, are very similar, there's some fine differences. For example, both kits contain a fully optimized flow panel, while the PBMC kit contains 8 antibodies, the whole blood kit already contains 9 antibodies. And the PBMC kit also already contains 1 SARS-CoV-2 peptivator of choice. Both kits contain cytostim as positive stimulation control. And of course, both kits also contain all the required buffers and reagents and have a very high capacity. So here on the top, you can see the workflow of the kit. So after isolation and stimulation of the cells with the SARS-CoV-2 peptivator, for example, the cells are then stained with the flow panel included in the kit. This is also shown here in the table on the bottom. And then the cells are analyzed basically on any standard flow cytometer, like, for example, our Maxcon analyzers. And it's also good to know that you can create your own stimulation mix by combining different peptivators. And as you can see here in the plot on the lower right, you can see that both SARS-CoV-2 reactive CD4 and CD8 positive T cells can be detected in just one sample through a combined analysis of activation markers and cytokines. And we are currently also working on additional versions of the kit, and they will be released very soon. However, we also offer other really convenient, ready-to-use flow analysis kits, such as, for example, rapid cytokine inspectors, which allow rapid multi-parameter analysis of activated human CD4 and CD8 positive T cells. Also, our MaxPlex cytokine kits available for both human and mouse samples, and they enable a multiplex analysis of several soluble cytokines in one sample, and they also work on every standard flow cytometer. And last but not least, the so-called 8-color immunophenotyping kit, which enables a optimized flow-based evaluation of various leukocyte subsets, such as monocytes, neutrophils, eosinophils, T and B, and NK cell populations. And of course, all of these kits are actually compatible with our so-called express modes that are available on all of our Maxcon analyzers. And you can imagine these express modes as some kind of software tool that will help you to automate your flow analysis. So finally, we'll take a short look on our very powerful cell sorter, the Maxcon Taito. So the Maxcon Taito really revolutionized cell sorting by offering some very unique advantages. And it is definitely the ideal sorter for infectious samples. So for example, the Taito enables really safe cell sorting as the sample stays inside a closed cartridge system for the entire aerosol-free sorting process. Furthermore, the Taito is really gentle to your cells, which makes the cells really perfect to be used in any downstream application. And also the full sterility of the entire sorting process make the Taito the ideal instrument in the context of, for example, translational applications. And here the Taito is already part of several clinical studies involving, for example, engineered immune cells or CAR T cells. And another key advantage of the Taito and also our Maxcon analyzers is their small size, which actually makes it possible to place these instruments directly into a flow cabinet. And let me just briefly point out that several Taitos are already being used in the context of SARS-CoV-2 research, here, for example, for the isolation of SARS-CoV-2 specific T cells or also B cells. So we know actually that the Taito is a really perfect solution for the gentle sorting of rare cells from, of course, not only, but also infectious material. So finally, here you can see a schematic overview of our whole antigen virus-specific T cell portfolio and also the entire workflow. And as you can see, we actually cover the whole workflow with products for sample preparation, cell culture, cell separation, and cell analysis. So to sum up, the most important message of today is that we will manage this crisis together. And there are a lot of benefits that come from a comprehensive virus-specific T and also B-cell research. For example, clarification of highly relevant questions in regards of reinfections, vaccines, protective immunity, or the role of cross-reactive cells. And furthermore, we can analyze these cells to monitor long-term protective immunity, be it after natural infection or also vaccination. And this will then hopefully lead to better understanding of the antiviral immune responses, a better disease prognosis, and hopefully also better treatment options, and also a faster return to a more habitual, more normal life. So by now, I hope I have convinced you that Miltenyi really covers the whole antigen virus-specific T cell workflow to support your research. So before we end, please acknowledge the displayed disclaimer. And with that, I would like to close and with this nice picture of our company campus in Bergisch Gladbach at night. And I would like to thank you very much for your attention.